Across every field and forest, plants build walls from interlaced sugars. Cellulose gives those walls strength, but hemicellulose fills the living spaces between. It curls around fibers like a flexible net, binding stalk, husk, and seed. In dry straw, fruit skins, and wood chips, this hidden net remains. For people, hemicellulose was long a locked storehouse of useful plant matter. Water alone cannot easily loosen it. Heat and force can be wasteful. The wall resists because its sugars are branched, crossed, and quietly tangled. Yet in nature, fallen leaves never remain locked forever. Fungi thread through litter, tasting the wall and opening it piece by piece. Bacteria in soil and compost share the same patient craft. Inside grazing animals, microbes turn grasses into nourishment through quiet chemistry. Termites, too, carry tiny partners that help unlock wood. The tool behind much of this unlocking is called hemicellulase. It is not one blade, but a family of small, precise tools. Each member fits a different link in the hemicellulose web. To understand its history, we begin with plant chemistry in the 19th century. In 1838, Anselm Payen separated cellulose from plant tissues with unusual care. His work showed that plant walls contained more than simple woody matter. Later botanists noticed other wall substances that behaved differently from cellulose. Ernst Schulze helped give these related materials the name hemicellulose. The name suggested half cellulose, though the chemistry proved far richer. Researchers studying germinating seeds saw cell walls soften during early growth. They suspected living tissues made invisible agents that dissolved wall reserves. At the time, many such wall opening activities were grouped under sitase. That broad word held early clues to what hemicellulases would become. As microbiology matured, molds and bacteria entered the story more clearly. Cultures growing on bran and wood released substances that thinned plant gums. Scientists learned that xylem, a major hemicellulose, could be cut by enzymes. Other enzymes attacked mannans and arabinins, revealing hemicellulose as a mosaic. The single name hemicellulase gradually came to describe this cooperating family. A practical path opened through fermentation traditions already rich with molds. Koji molds, long used in East Asian foods, became valuable research organisms. They produced many enzymes at once, including helpers that loosened plant walls. By the mid-20th century, fungal fermentation supplied crude hemicellulase preparations. Researchers refined the idea by separating xylanases, mannanases, and side branch cutters. The enzyme family became especially important in the pulp and paper world. Wood pulp carries hemicellulose fragments that hold colored lignin in place. In the late 20th century, researchers tested xylanase before chemical bleaching. The result was a gentler opening of fibers before harsher steps arrived. Nordic forest laboratories helped show that enzymes could reduce bleaching burdens. This was a turning point for hemicellulase as industrial green chemistry. Microscopically, hemicellulase works without tearing the whole plant wall apart. It lands where its shape matches a tiny section of sugar chain. Then it adds the chemistry of water to separate one link. A long branch becomes shorter, and the wall relaxes slightly. Millions of such small moments can change the texture of plant material. In bread making, hemicellulase helps dough handle water held by grain fibers. The dough can become easier to shape, with a softer finished crumb. In fruit processing, it helps release juice from cell wall networks. Clearer liquids can emerge when suspended plant fragments loosen and settle. In animal nutrition, it helps make plant feeds more accessible to digestion. The enzyme does not replace the animal, it supports microbial partnership. In brewing and cereal processing, it helps manage sticky grain components. In textiles, related hemicellulase actions can soften natural fibers gently. In biomass conversion, hemicellulase helps expose sugars from agricultural residues. Straw, husks, and bagasse become more than waste after harvest. They become sources for materials, fermentation, and renewable carbon pathways. The promise rests on using mild tools where brute force once dominated. Hemicellulase reminds us that softness can be a form of power. Its history runs from seedling observations to forests, farms, and factories. It began as a mystery inside plant walls, nearly invisible to chemistry. It became a working language between biology and industry. Today, each careful cut can save heat, chemicals, water, or waste. The gains may be quiet, but they accumulate across countless processes. In a future built from renewable matter, such quiet catalysts will matter deeply. At Enzymes Bio, scientists and enzyme enthusiasts work to make this biology easy to reach. One platform, many enzymes, no tangled procurement, for research labs and growing businesses. A green technology with a long future. Learn more at enzymes.bio.